Excellent. Thanks a lot, Marinka and Europe, for inviting me. Yeah, so what I will talk a bit about is how we can use data to repurpose drugs. And the emphasis is here more on data. So how can we actually use particular types of data in order to get a signal to make decisions? And the emphasis right now is quite often on algorithms. So we have the age of AI right now. But the question is quite often, where's there actually a signal in the data? So what is the right biological data to use? Is it sequence data, gene expression data? Should we do imaging? What should we actually look at? Yeah, and in my opinion, that gets underemphasized or neglected a bit right now. And so I will discuss some of the approaches that we have right now, which certainly bring some opportunities, absolutely, but also some of the pitfalls and areas for possible improvements in the future. So any statements I make during my talk and my capacity as an academic at the university. So where are we right now? So we are at the third wave of computers in drug discovery. So the first wave that was in the early 80s. So here I dug out an old cover from Fortune. So that was the age when computer graphics came up, docking, for example. So dock was published in 1982. Silicon Graphics was founded in 1981. And so that's the Fortune cover, the next industrial revolution, designing drugs by computer at Merck. That was a company that was a really big driver in that field. So that was in the early 80s. Then in the 2000s, some of you might remember that was the biotech bubble. Virtual screening was very popular. For example, as a student, I joined a startup as well in that field in Berlin. And so that was the second wave. And today we're in the third wave. And so here are some recent headlines from that area. So how artificial intelligence is changing drug discovery and so on. And certainly it's not exactly the same as before, but many technology developments, they come in waves, right? And so they come and then they go a bit, and then people figure out where can technologies actually be used and so on. And so right now is the third wave. And so I would like to ask the question, how can we realistically assess actually how good are we right now? What is needed for proper validation of the methods we have, and which types of data do we actually need to make decisions? I cannot answer all those questions, but I would like to emphasize how important those questions are. Yeah, so I'm old enough to remember the 2000 biotech bubble. That was also the time of the Human Genome Project. So that was published in 2001. And that was a very important project because we have a reference genome. We can catalog genomes or genes, for example. Before, I still remember those speculations, how many genes the human actually has. And that number was frequently overestimated and was lower than expected then. So what was published around that time? So that was the biotech bubble, so the second one of those waves I described before. And so what was proclaimed around then was an article here. The number of drug targets will increase due to the Human Genome Project by at least 1 order of magnitude, and target validation will become a high throughput process. And so the expectation was that we will have more drug targets, so 3,000 or even 10,000 targets compared to, at that time, 483. And well, so more recent article in Nature Reviews Drug Discovery estimates that current, or at that time current, number of drug targets would be around 667. So as we can see, nearly 20 years later, we certainly not reach what was proclaimed at that time, but still the number of drug targets increased by about 50%, and that certainly was something as well. And so, as we can see, if you're in an age where lots of things happen, it's easy to overestimate the impact. But what some people say is the short-term impact is quite frequently overestimated, but the long-term impact underestimated. So who knows, maybe that's the case here as well. Certainly, we need to analyze data, but we need to look at data in combination with representations of data and the method we use as an integrated system, so to speak. So that's the main part of my talk. So I will go through, in the first part of the talk, go a few examples, how we can repurpose compounds. So which types of data can be used? Where does that work? I will talk about approaches using gene expression data for that purposes, to select individual compounds, to also select compound combinations. One of the applications will be to infectious diseases as well, namely to malaria. And then the second part of the talk will be on data and drug discovery. And that's something I could describe as a painful relationship. Unfortunately, we quite often don't see the signal in the data that we have to the extent we would like it to have. And so there's still possibly some development needed. So I will discuss aspects of the data we have, how difficult prioritization is, for example, in knowledge graphs. So is our data good enough? What do we actually require to be able to traverse or mine a knowledge graph efficiently? How do we validate right now computational approaches? Is that sufficient to say that there's really progress? And I will also interweave this theme of technology push versus scientific pull now and then. Because as we've seen, the Human Genome Project that was finished in 2001, but right now, because we in the age of GWAS studies, for example, in some sense, there's still the echo of the 2001 Human Genome Project, right? That's a new technology that came to play, and that is still being analyzed. So what do we see on that level of information actually? On the genomic level, what does it tell us about the individual propensity for diseases, for example? Okay, so repurposing, what can we do? So what works, or works in some case studies at least? So this is the worldview, so to speak. I'm usually trying to communicate to my students, and that's an integrated view, or life science view, I should say. So that includes the molecular structure. So that's the independent variable in our system, so to speak. We can synthesize most of the molecules we want to synthesize. We can store chemical structure reasonably well in the computer. Sometimes stereochemistry is difficult. We can have mixtures and so on, but still we can handle that reasonably well. But what we also need to look at is the protein or mode of action angle, because that helps us understand what a compound does in a living system, as well as to select compounds. So that's a very important mediator between molecular structure and the effect on a phenotype. And the link between molecular structure and phenotype is crucial, and because that's what we actually want to cure. We don't want to cure a protein. We don't want to find a ligand for protein. That's only means to an end. We want to change the phenotype, of course. And the nice thing about this, well, to some extent, simple view of the world, I will come back to that later, is of course we have data that links the different corners of that pyramid. So we have bioactivity data that links molecular structure with proteins. If you go into PubChem, hundreds of millions of data points, for example, many are inactives, of course, because the typical ligand is inactive against the protein, but we still have millions of active data points as well. So that's relatively data rich. Then we have pathways that links proteins to phenotypes. It's in quotation marks here because that's maybe more human view. Humans like to put things into drawers and to categorize things. But of course, biology is more reconfiguring itself. It's maybe more a network and even more dynamic than that maybe. But we have some type of ability to link proteins to phenotypes. And finally, we also have phenotypic response data. So we know we can measure what a compound does to a phenotypic system with all the problems that brings. For example, capturing all the variables of the system. What's the genotype of the person that chose an adverse drug reaction and so on? Often we don't know that. But still, we have lots of data that analyze that triangle or pyramid. And so we can ask questions. And usually if we have data for two, we can ask for the third. So we can ask the question for the mode of action, for example, and that's the area of target prediction. So we can understand how a compound works. We can also ask the question, if I want to modulate a certain phenotype, what are the pathways I should modulate? So what are the kinases, for example, in that pathways I should inhibit? So what is the pathway-focused library I should screen, right? So we can go back to chemistry. Or we ask the question, how do patients respond to a treatment of a drug? The typical example is HIV. It's very cheap to sequence HIV, the viral genome in a patient. So I can say, I have that particular mutation in reverse transcriptase, so I should give that or that drug, right? So we can integrate that information we have in that triangle and ask questions, and very practically meaningful questions, and answer them underlined by data we have. And so one way to use that is using gene expression data in combination with target predictions and pathway annotations for repurposing. So in that triangle, we had this mode of action layer, right? And in this case, that is represented by gene expression changes and is linked to the compound side, because a compound can cause gene expression changes in cells. And that gene expression is also linked to a disease side, because you can measure, if you have diseased tissue, what are the gene expression changes that possibly drive that particular disease phenotype. And via this gene expression level that's represented in the middle here, we can link the compound level on the left, as well as the disease level that's represented on the right. And that was published in 2006 first with connectivity map. There were hundreds of papers of many groups that tried to use the gene expression based repurposing principle. The idea is quite simple. So you have upregulation of genes in a disease. So the compound should induce downregulation of the gene to get back to the healthy state. It's probably a bit simplistic to think about that in the mechanistic way, but from the signal detection side, that works surprisingly well. So we're able to pick compounds that in many studies were found to really modulate the diseased phenotype. And this is what we did as well. So for example, to differentiate stem cells. And so this is the work of Yazaman, Kalanta, Motamedi. The reference is given here. And so what are the data sources we used? There are two main data sources out there, connectivity map and links. So that's in one case Affymetrix chips. In the other case, it's very short 1000 gene expression readouts. These are public data sources from the broad. And so I should say gene expression data is probably at a sweet spot when it comes to data generation. It's quite cheap to generate such data, especially with technologies such as DrugSeq and so on. It's relatively cheap to do that. And it, in our opinion, contains probably sufficient signal for signal detection, for repurposing, for example. But what's more problematic is to model that data. So if you want to have dose-response relationships, for example, then that type of data is much more tricky to handle. But still, if you balance the cost, for example, and the signal you get from your data, that is quite a good deal, in my opinion. But still, it's a love-hate relationship, if I might call it like that. And the reason is that if you see something, then that is great, but it all depends on the setup of the biological system. Which cell line, dose, time point do you have, for example? And not in all cases you see a signal in gene expression data. Yeah, and so in some cases that is actually a tricky system to deal with. But still, in some cases it clearly works. And one of those case studies is shown here for repurposing of compounds for differentiating stem cells. And so differentiating stem cells, so what you do is you're taking a stem cell, you apply a compound, and then you see over a process of, in this case, 3 weeks, so 21 days, you can track the cell differentiation. So you can characterize that using gene expression data, proteomics data, in order to establish how pure the cells are that you get after the differentiation protocol. And what you see here in this particular case is that after 21 days, you actually get pretty pure cardiomyocytes. And this is one of my favorite papers, I think, from the last many years as a group leader, because it really shows the combination of chemistry, biology, as well as computer science and algorithms, how powerful that is. You put in a stem cell, you apply a compound, after 3 weeks, you get a beating cardiomyocyte. So you can, this is only a static picture, but I've also seen a movie, so you get a beating, heart cell effectively, okay? And that whole process is driven, of course, by gene expression changes. So that is one area where it is quite, understandable that using gene expression data to drive such a process is actually working. We can also use that in other areas, for example, for selecting compound combinations against pancreatic cancer. So in this particular case, if we repurpose compounds using gene expression data, we want to invert the gene expression profile of the disease, right? So gene gets overexpressed in the disease, we want to downregulate the gene. But if you look at the gene expression signature as parts of that signature. So you have a correlated part between the disease and compound treatment, and that's the one you don't want to have, right? Because the compound would upregulate the genes that are also upregulated in the disease. And you have an anticorrelated part, and that's the desired part, right? So you have upregulation in the disease, and they want to downregulate that with a compound. And so the hypothesis of selecting compound combinations was here that the anticorrelation of both spaces between disease and compound is desired, but the correlation between spaces is to be avoided. And so you combine compounds in a way you get the highest possible anticorrelation and the lowest possible correlation. Or in other words, the sum of all compounds should then revert the disease gene expression profile back to the healthy state. And so this is what we did with people in Edinburgh's. And so there's one treatment for pancreatic cancer, and that's gemcitabine. The problem that's here shown on the right-hand side. So what we have here are gene expression profiles mapped onto pathways for a series of compounds that are known partially to be active, against pancreatic cancer cell lines. Can you actually still see me here?
We can see your screen, but cannot see your picture.
Okay. Yeah, it's just the menu disappeared. Okay. Yeah, I think now it works again. Okay,
my apologies.
Okay, very good. So we had, I'll just go back one slide. Yep. Okay. So we have a gemcitabine, and that's a treatment that's used in the clinic against pancreatic cancer. The problem with that is it's not very efficacious. So you don't get even a value where you kill 90% of the cells. So you don't have an EC90 value because it's simply not efficacious. It does not work well enough. So the question from the clinical side that we ask ourselves is, what is the combination of gemcitabine with the second compound that we should use against pancreatic cancer? And so what you see here is the gene expression changes that are induced by pancreatic cancer, by the PANC1 cell line on the right-hand side, as well as that of treatments that are used, or compounds that are known to be active partially against that particular cell line. And you see that in many cases, you actually have a pretty good inverted gene expression profile. So this is this anticorrelation of gene expression signatures for repurposing I referred to. So you see that you have an inverted profile of PANC1 compared to the treatments, for example, also gemcitabine. But if we look into that in more detail, we see that also correlated parts of the gene expression signature are present. And so what we were setting out to do was to combine gemcitabine with the second compound. And this is still something we are writing up, so I cannot show details here, but with the second compound, and we were able to find synergy of that clinically used compound with another approved drug to see, to exploit, to show synergy in the PANC1 cell line, to show increased efficacy in the clinic. Okay, so what does that mean? We can use gene expression data together with algorithms to select synergistic compound combinations. And we did that also in malaria. So in this case, we used ligand target predictions, information from combination screens. We mapped that on pathways and to select novel synergistic combinations. So that's shown in detail on the next slide. But the point I'm making here is that we use different types of technologies, predicting protein targets for ligands, experimental information from combination screens, pathway annotations, and then we used all of that to select compound combinations that are synergistic against malaria. And so this is what the workflow looked like. So we selected individual active compounds using gene expression data from patients with severe malaria versus mild malaria. And so this was, and you could argue that what the rationale for that was, because that was host gene expression data, right? But we followed that protocol that was described here. You can, of course, vary that for very good reasons. So we selected in this way single active compounds based on gene expression data. We enumerated the target predictions. So we knew the compound inhibits replication of Plasmodium. We predicted targets. So we knew which pathways and combinations we were able to target with the compounds we selected. And then we had an experimental combination screen, and we knew which combinations of compounds showed synergy in that combination screen. And for those compounds, we could also predict targets and annotate with pathways of synergistic combinations. Okay, so from both sides, we knew which pathways we were able to target, and we knew from the combination screen which combinations of pathways led to synergistic compound combinations. So we can then combine the new compounds we selected and test in which cases we actually see synergy in experiment. And that worked remarkably well. So we were able, against different strains of Plasmodium and so on, to achieve a relatively high precision and recall and synergy over the background distributions. Yeah, so we can also apply that concept to infectious diseases. That's the point here. And so that tells us that we can use biological data as well as chemical data, target predictions, for example, in combination with algorithms in order to select compounds, repurpose them, select compound combinations, right? But there's still some open questions, as I alluded to in the beginning. So what are those? So in the simple worldview that I illustrated before, we have a compound that cures a phenotype, cures a disease, for example, and a compound that binds to a target. But the problem is that all of that data is, of course, quantitative, right? So we have an IC50, for example. Then the next question is, if I have an activity against the GPCR, that might induce different downstream signaling, beta-arrestin signaling versus G protein signaling, for example, leads to completely different adverse reactions in many cases, and so on. Pathways are very heterogeneous in their annotations. So what's actually a pathway? Yeah? Is the protein-protein interaction what in a yeast system? What does it mean in my particular disease state, for example, right? So this is all much less clear-cut than we would like it to be. And the important thing here is probably at the bottom of the slide. So we label often what we can measure. That's the technology push. So we assume now that we can sequence cheaply, let's sequence, and this is probably where we find the signal, but there's only an assumption. Yeah, and in some cases, cancer, for example, there might be stronger links. In some other cases, there are much weaker links. Yeah? So always distinguish, is it a technology push or a scientific pull that leads us to using particular types of data? Yeah. So the labels we have, are they directed? Because that's what we would need for reasoning, right? Are they causal or only association based? That's quite often the case. What is the confidence in different types of data? Is it quantitative data? Yeah, we need to answer all those questions in order to reason with the data we have. And this one example here, so ketamine. So the point here is we have one compound. Ideally, we would like to link it to targets and indications, but the real world is different. So that's a compound we've known for 50 years. In green, you see here the indications it's used for. So it's an anesthetic, a street drug, antidepressants, bronchodilator, and so on. And some of those indications are still being discovered right now. So these are the indications, right? It's a list, it's a long list, and it's growing. Then we have different doses we can use. Here it's IV. If you use it at 2 mg per kg, that's when it's used as an anesthetic. If it's a bronchodilator, it's a much lower dose. It's only 0.5 mg per kg, for example. Then how do we annotate with targets? What's the mode of action of a compound actually? Because not every target of a compound corresponds to the cause of the effect, right? So it's not necessarily the mode of action. And so that is not clear in this case as well. There's the NMDA receptor hypothesis, opioid receptor hypothesis, and also metabolites might be active, right? So it's very difficult, that's the point here, to annotate a compound with an indication as well as a mode of action. That's in many cases not quite clear. Another case in point, adverse reactions. So in a simple case, you would like to ask a question, so does my drug cause an adverse reaction? Yes or no? Okay, so I have two entities, I have a link, and that's it. But in reality, that is much more complex, right? We have a patient with this genotype is unknown. The disease endotype might be unknown. So a disease can have multiple subtypes, right? And that patient takes a particular dose of a drug. And then we see an adverse reaction in 50% of the cases, but with different severity and so on, right? And only in males and only after a certain time, for example. And the question is basically, how do you put that information into your, for example, knowledge graph? And that is much more tricky to do. So if you look at this list, it's much more difficult to answer the question. So does my drug actually cause an adverse event, right? It depends on so many other variables as well. So mining this type of data is actually very, very complex if we really consider the difficulties in clinical data, for example. So how are we meant to navigate in spaces that are so poorly annotated? So we have hundreds of thousands of entities, millions of edges, tens of millions of possible novel links. And it comes from very different sources with a very different meaning. And often it's not quantitative, not directed, and not causal, but that's what we would like to have, right? So how do we prioritize in that space? Because we want to pick individual compounds and we have 10 million links. We can link nearly every drug to every other indication, right? That doesn't really help us to prioritize. And usually in practice, what happens quite often is you need to have an expert in the loop. So I'm a strong advocate of that. There was a discussion before, an expert in the loop to make sense of that. But we need, in combination with that, I think we also need much better data. And that does not, it's not good if it's only a technology push, right? We need to ask the question, so what is the data we need in order to draw meaningful conclusions? So it needs to be driven more from the scientific side, in my opinion. And so to prioritize in such a complex and heterogeneous data system is often not as trivial as plugging in the data and running an algorithm. Since we need to look more at the data and what the data, individual data points actually tell us. So I was writing that up. So I was writing a blog here, this drugdiscovery.net recently. And so this is the link to the blog entry where I discussed the importance of data in data mining using AI in the life sciences. And so this is the pyramid I was coming up with. I'm certainly not the first one, but the point is here that the question or the hypothesis at the bottom of it, and that tells us, so the question tells us, okay, we have diseases that are driven and cause a change in gene expression. That tells us we need to generate gene expression data. We need to represent it on the individual gene level, pathway level, and that defines which method to use, right? It's a pyramid, and that starts ideally at the hypothesis level. Quite often, though, we start at the data level. So we take the data we have, and then we start answering questions, right? But this is not really the basis where it all starts. So there's lots of attention currently on methods, also representations, and that's good, but we should not forget also what is the question, the hypothesis? Do we have the right data? Do we have the right data to actually answer the question that we ask? Yeah, science pull versus technology push. And there are two very different types of data we can have. And that can be early stage data, usually in vitro data. And that's usually where lots of the AI that we do right now in drug discovery is happening. early discovery data, but it's often proxy data. So what does that mean? We measure binding to an individual protein, for example, in the biochemical assay. We measure solubility, and we measure cytotoxicity against the cell. Yeah, that's easy to measure. We can generate it on a large scale. It's good for data mining. It's not so good for in vivo patient relevance. That's on the left-hand side of the slide. On the right-hand side, we go into the area of efficacy and safety. That's usually in the area of in vivo data, much more quantitative data. Everything depends on dose, right? Even water is toxic. If you drink 3 or 4 liters of water in a very short time, even that is toxic, right? So it needs to be quantitative data that we have. In this case, we have more complex models though, because we have multiple endpoints, histopathology, for example. Yeah. And so in this case, animal data is also much more costly to generate. So on the right-hand side case here, we have much less data, more complex data, and that's much more difficult to handle from the machine learning angle. We also have many more confounding factors. For example, if you have a male handler of animals, of rats, stress levels are higher, that can confuse your experiment, okay? And you don't have that if you have biochemical assays. So we can either operate in proxy space, that's quite often where current AI and drug discovery happens, or in efficacy or safety space. And this is usually more in vivo relevant, but we have much less data, right? And that is a problem. Now, those are the questions we ask in both spaces are usually different. So in early discovery space, it's more discovery setting. Give me any answer, any compound out of the many possible active compounds out of millions of compounds, right? We have many possibilities to be right. But if we're in the efficacy and safety setting, we need to predict for a particular data point quantitatively, and there's a long list of criteria to roll out any type of toxicity, for example, based on limited data, right? And that is much more difficult to do. So we should need to shift, in my opinion, from the left-hand side of that process, operating in proxy space, in order to go to the efficacy and safety space to really do quantitative predictions. Yeah, and that also relates to the validations that we see right now. So this is how a drug operates, right? It has an effect on a protein or an organ or on the membrane, but there's some effect on the biological system. But it also has PK properties, metabolism, secondary targets, and so on. And that causes the physiological effect. But what happens in practice quite often, because of this data-driven question setting, is that AI and drug discovery quite often operates on the protein level. And that is more the ligand finding level and not really the drug discovery angle due to the data. So you look for the key where the light is more or less. So you're asking questions where you have data. That's not necessarily the right thing, because what is missing there, of course, is in vitro to in vivo translation. There, going from a target to safety and efficacy, that is key. So what do we really validate? And that's the important aspect here, because how we generate data, that influences what we can validate afterwards. So what do we really validate if we talk about AI and drug discovery? And that's a more recent blog post, which you see linked here at the bottom of the slide, that aroused some interest. So what do we actually validate, and how do we validate properly? Do we want to discover ligands or drugs? And what do we need to do in every case to really show that our compound is efficacious? We need to have a meaningful baseline comparison. If you do an experiment, you have a negative control, you have a positive control, right? In order to put what your new compound does into context. Yeah, but what are those baselines if we have models? Yeah, so you need to have some kind of mean predictor and a state-of-the-art model also to put the performance of your new model into context. But that's a problem. Validation is a problem, because if you have models that involve networks, yeah, quite often you have a connectivity bias. You have nodes that are much more central, and there's a bias. It's difficult to evaluate your model in an unbiased manner due to this connectivity bias. The similar things exist in chemical space, of course, because of many analogs, for example. If you do prospective validation, which is by itself a very good thing, the problem is that there's often, and I would even say always, some manual bias. And this is a very good quote. That was my favorite quote from Steve Kearsley's recent article. He's now at Google. Model validation is always process validation. So what does that mean? You cannot validate a model by itself. If you do retrospective validation, you do cross-validation or so, but you don't predict anything new, right? You sample the same chemical space effectively. But if you do something prospectively and you pick new compounds, which is a very good thing to do by itself, then you need to select them, for example, from a vendor library. Yeah? But that depends then on availability of the compounds, for example, right? And is that then model validation? No, it's validation of the whole process, right? So it's very difficult or nearly impossible, I would argue, to validate a model in our area, drug discovery area, and by itself. Yeah, model validation is always process validation. It's embedded into how you use the model. So summary of my talk, key learnings. Computational models are only as good as the underlying data. We can and should apply algorithms. Of course, we should analyze the data we have, chemical structure, bioactivity, gene expression, and so on, for example, for repurposing. But we shouldn't fool ourselves. Coverage of data, for example, is tricky. Predictivity of data, so which type of data, especially in the biological domain, contains a signal. How do we validate so that we really validate not only on a biochemical level in the drug discovery context? And that is very tricky. And a method follows the representation of the data, and that follows the data itself, and that needs to follow a hypothesis or question. If you do hypothesis-free data analysis, yeah, and you start at the data level, that can be tricky. I worked on high content screens before. That is not the right scientific approach. So let's get a step. Let's step a little bit back and see what is actually the hypothesis, the question we want to ask to generate the right data. And I think what we also need is to identify which readouts matter. Probably I didn't allude to that, but we need consortia not only for data sharing, but also for generation and agreements on best practice in data analysis to make sure what we do is reproducible. Okay, thank you for listening.
Great, fantastic, Andreas. Thank you so much for bringing the complexity of data to our attention and for discussing the kind of this discrepancy between technology proofs and scientific pool. So Eugenia Fava is saying, I can't agree more with the importance about data. However, many data sets out there are often badly curated, not reproducible, and so on. In your opinion, and you touched upon this at the end of your talk, how can we as scientific community, what should we do to make better data available more easily?
Yeah, I think we need to properly talk to each other, people from the, let's say, pharmacology, drug discovery domain and the computer science domain, for example, so that we have data that's in a format that can be mined easily. Yeah, but also where we have all the relevant annotations, for example. For example, this quantitative information, that is really key to have information on which dose causes which type of effect, for example. The compound is not toxic, yeah? But that is, we need to have something from the practical relevance side, all the annotations, right? As well as the computer science view, so that we have in the right format, right? Interacting in this way, I think that is really very, very important.
A follow-up to that, what about doing validations with causal inference studies or population level data such as GWAS catalog, epigraph database, or other more causally determined measurements?
Sorry, Bob. Okay, no, I'm not quite sure I understood the question. Sorry, what was the question for?
What about validation using causal inference studies and population data like GWAS catalog?
Yeah, what about using it? Well, I mean, it depends on how you use the data, right? I mean, the point is basically, how do I go from associations to establishing a really causal relationship? And that is a key question here, because only then it's actionable. I need to know, can I modulate my protein to reverse a diseased phenotype, right? That's what I'm interested in. It's not just an association, because that doesn't tell me if that is enough to really causally modulate my protein of interest, right? It depends on what you do precisely. Yeah.